Okay, so in this next video, we're going to talk a little bit about some complications that we see in Mendelian inheritance, where it's not quite as simple as dominant and recessive. So remember, Mendel wrote his rules and came up with his ideas way before we had any idea about chromosomes, DNA, genes, even proteins. And so he was explaining things using terms that made sense to him in what he could see at his level from the whole plant kind of view. But now we know more about what's going on at the molecular level with proteins and genes. There's a little more complexity. So the first example is the idea of incomplete dominance. And that's what this slide is talking about. So this is a classic example. Here are two flowers. Luckily, when Mendel did his work, he used peas, and peas were either purple or white flowered, and there was no weirdness going on. And so it was simply purple or white, and purple was completely dominant to white. So in Mendel's example, I think if you remember from before, a plant that was big P, little p, had purple flowers, and they were as purple as a plant that had big P, big P. All right, that's not the same in every single kind of plant. So this is a different type of plant. This diagram here is showing snapdragons or antirrhinums, which are a kind of flower. Here are carnations. Those are the kinds of flowers that you have like in lapels, you know, for corsages and stuff like that. All right, so in this situation, we're going to cross a red flowered true breeding plant with a white flowered true breeding plant. And we're going to say the C gene is responsible for making a protein that goes into the petal cells and gives them a color. And the C gene comes in two versions. It comes in the red version, color red, and it comes in the color white version. So what this means is this is an allele that makes a protein that folds up and looks red. This is an allele that makes a protein, and when it folds up slightly differently, probably because of a mutation, and it looks white. All right. But if you look at how I wrote those, I used uppercase for them both. There wasn't an uppercase and lowercase one. So in this example, we're going to take a true breeding red flowered plant. So that's going to be color red, and we're going to breed it with a true breeding white. So color white. And if we do a Punnett square, which is what we would normally do to find out how the babies would come out, we're going to get CWCR, 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 right? Now, if this was a normal relationship between the two alleles and one was dominant, one was recessive, these flowers would either all be red or all be white, whichever was dominant, right? But that's not what you see. So these are, all these babies in here are all heterozygote. They're all CWCR. But when you look at them in real life, they actually look pink. They're not red. They're not white. They look somewhere in between red and white. It's like we've created a new allele, CP for pink, right? But we haven't. We have not made a new allele. What's happening here is if you can imagine that each allele is responsible for making, say, 1,000 molecules of protein in the petal. So if you have the CR allele, you can make 1,000 molecules of red. And if you have another CR allele, you can make another 1,000 molecules of red. So you're making 2,000 molecules of red. And that's why these petals look so very red. This one is making 1,000 white proteins and another 1,000, so it's making 2,000 white proteins, and it's looking super, super white. If you put them to breed them, have them together in an individual, like this offspring here, these cells and these petals are making 1,000 red protein molecules and 1,000 white protein molecules. It's making both the proteins. It's making the red and the white, and they're both going into the petal cells. And so what happens when our eyes look at this, you see the red and the white together, and our eyes kind of pick it up as pink. Because to us it looks like it's merged together. It's like kind of, if you could zoom in on these petals at a molecular level, you see tiny red dots of protein molecules and tiny white dots of protein molecules. They'd both be present. But when you stand back and look at it, it merges together and it appears pink. Just like how printing in an old comic or newspaper used to be lots of tiny dots, but when you stood back, it looked like a color. So in this pink flowered plant, red isn't dominant, white isn't dominant. They're both there, they're both being expressed. It just appears to our eyeballs like they're mixed together, they've blended and they've made it pink. Now, as I say, that's not what's happened. It's just that our eyes can detect both colors, and both white and pink at the same time, and to us, our brain detects that as pink. And we call this incomplete dominance. It really is, there's no dominance and there's no recessive. They're just both there. Both those proteins are there at the same time. But unfortunately, we have these terms of dominant and recessive that we have to work with. And so that's how we've termed this. So, the hallmark of incomplete dominance is that it looks like it's blended together, that the alleles have blended together in the phenotype of the heterozygote, but they haven't really. And then in this example, on this diagram here, they've done a Punnett square where they've crossed two pink flowers. So a pink mom, which would be CRCW, crossed with a pink dad, so CRCW. And this is going to be a CRCR offspring, and this is going to be a CRCW. This is going to be CRCW, and this is going to be CWCW. And here you have one red, two pinks, and one white. Right? So you have 1 to 2 to 1 ratio of red to pink to white, because in this example, the heterozygous individual actually has a different phenotype because it has its own blended phenotype of the two. It looks like it's blended. So that is incomplete dominance. Both alleles are expressed. To our eyes, it looks like they've blended together even though they haven't.